Hey everyone, it's Jamie Batts, your instructor for AMP2. We're continuing on with chapter 20. We just finished this section talking about lymphocytes. Now we're going to move on and talk about the lymphoid tissues themselves, get more specific about them, and I'm going to go into some details. So let's go ahead and switcheroo right like this. That's where we left off. Let's talk about lymphoid tissue. So we have different types of tissue in our body we determine as lymphoid tissue. It's basically connective tissue that has a high concentration of lymphocytes. That's what lymphoid tissue is, right? Connective tissue with lymphocytes. There are lymphoid nodules. These are densely packed lymphocytes within areolar connective tissue. The nodules cluster together. They form large masses. There's no fibrous capsule around them. It's just a cluster of nodules. That's all it is. Or a cluster of lymphocytes, I should say. The lymphoid organs themselves are separated from surrounding tissues by some type of fibrous capsule. So there are aggregated lymphoid nodules, which we call Peyer's patches. These are within the linings of the intestine. So they're basically clusters of lymph nodes that are deep in the epithelial lining of our intestines. They have a special central zone called a germinal center, and this is where the lymphocytes are dividing. Remember, any place in your body where you have like the potential for foreign invaders to come in, you're going to have a high presence of lymphatic tissue there because you want to protect the body from any invaders coming in. You also have MALT, mucosa-associated lymphoid tissue, within all of our mucosal linings. Our digestive, respiratory, urinary, reproductive tracts all contain MALT, and that MALT is just a high concentration of lymphoid nodules or lymph nodes there to gather any invaders that may be trying to come in. So here you see the intestinal lumen and kind of where that lymphoid tissue would be just deep to the mucosal lining. Tonsils are specific lymphoid nodules that are within the walls of the pharynx. Pharynx is the fancy word for throat. There's the pharyngeal tonsils, which are also called the adenoids. You might have heard of them before. They're located on the back kind of upper wall of the roof of our mouth area, just kind of behind the soft palate. There's the palatine tonsils. There's a left and right. This is on the back side of our oral cavity. This is what you can usually see when you open your mouth wide and the doctor says, oh yeah, I can see your tonsils, you know, that type of a thing. And then you have your lingual tonsils, which are just at the base of the tongue towards the back, more difficult to see. So there they are. The palatine tonsils are typically what you see when you open your mouth if you have some type of sore throat. So let's talk about those lymph nodes in general. So they're basically small organs. They have a capsule around them, and the shape resembles a kidney bean. They're typically small, about 1 inch in diameter. There are larger lymph nodes, like lymph glands, that are located in the neck, the groin, the axilla, your armpit area, and that's what you can feel when you have a sore throat. And your doctor might feel your neck and say, "Oh yeah, you know your glands are swollen." Right? The glands are the lymph glands, which are just very large lymph nodes in that particular area. These are basically little filtering stations. 99% of the pathogens that are in the lymph are removed from the fluid before it gets back into the bloodstream. Remember, the lymph are those little vacuum hoses, the lymphatics are the vacuum hoses sucking up all that extra cellular debris, any pathogens, etc., that might be in that interstitial fluid. So as it's vacuuming up, the lymph nodes are the little filtering stations which are going to check it before it lets whatever into the bloodstream, right? So let's talk about the path of the lymph, path through a lymph node first. There are afferent vessels. The afferent vessels are bringing the lymph into lymph node, and then there is an efferent vessel. The afferent is arriving, right? We've heard these terms before, right? When we were talking about nerves, right? The afferent vessels, and there's several of them coming into the lymph node, are kind of coming out from all different sides. The hilum is the indentation on the opposite side of the afferent vessels. So if you picture this kidney bean shape, that indentation on the kidney bean is the hilum. On the opposite side of the kidney bean, kind of the outer curve of the kidney bean, would be where all the afferent lymphatics are. So those are all bringing lymph in from the periphery into this filtering station. The lymph flows through the subcapsular space, and there's all these dendritic cells there. Dendritic cells are special immune cells. Students tend to think dendritic cells are some type of nerve cell. They're not. They look like nerve cells, which is why they're called dendritic cells. They have all these extensions and stuff, but really they're just a special type of immune cell. Then it flows through what's called the outer cortex. The outer cortex has lots of B cells. These B cells have germinal centers. The B cells can detect and kind of dipstick that lymph and say, ooh, we have some antigens here that we need to respond to. Once it gets to the outer cortex, then the lymph's going to flow through the deep cortex. The deep cortex are where the T cells are. So the lymph is going through a primary kind of surveillance with the dendritic cells. Then it's surveyed by the B cells. Then it's surveyed by the T cells. Then it gets to the medullary sinus where there's B cells and plasma cells ready to make antibodies. And then it flows out of the lymph node through something called an efferent lymphatic. The efferent lymphatic exits the lymph node at the hilum, at that little indentation. So what I suggest doing is taking this information and seeing if you can draw a lymph node based on these descriptions. It's in the book. It's probably on the next couple slides what an actual lymph node looks like. See if you can draw it out without looking. See if you can create that kidney bean shape with all of these afferent or arriving vessels, the indentation called the hilum, label the areas where the different types of cells are through the lymphatics, and see if you can understand the flow of lymph through the lymph node. Here's the thing. Think about it. If you have all of these afferent vessels and only one efferent vessel, do you think the flow of lymph through the lymph node is very fast or very slow? It's very slow, right? You have this traffic jam that's created. Imagine this major intersection, right, where you have all of these roads coming together. Everyone's trying to get in different directions, but there's really only one way out of that intersection, right? And so the only way out is through that efferent vessel, or by being gobbled up by a B cell or T cell, right? So there it is. That's what you should have drawn if you took me up on that challenge. You can see those afferent vessels. You can see the germinal layers where the B cells are. You can see the the outer cortex, the deep cortex, and the medullary sinus. And I love how this diagram here actually shows where everything is and labels it for you. It makes it very easy to understand. And you can also see how that efferent lymphatic is parallel to the artery and vein, which are going into the lymph nodes, supplying all of those B cells, T cells, dendritic cells with the nutrients that they need to survive. So let's talk about some clinical disorders associated with our lymphoid tissue. First, we have MALT. MALT is defending the epithelial and several tracts against any invaders that are coming in from the exterior environment. So any infection or inflammation of the MALT is going to basically represent something going on in the body. Tonsillitis, the inflammation of the tonsils. Appendicitis, inflammation of the lymphoid tissue of our appendix. So any swelling or inflammation in those areas is going to indicate that there's something going on with that general location, right, in the throat or in the intestines. You can stop the video here and answer these 3 questions real quick. Make sure you understand everything that we just went through. We're going to continue on and talk about the thymus. The thymus is responsible for producing thymosins. Thymosins are thymic hormones. These are fueling the T cell development. The size and secretory abilities with the thymus actually decline with age very rapidly. The thymus is very, very active when you're young, and then right before puberty, it pretty much stops, and then it's slowly involuting. In other words, it's becoming inactive. So by the age of 50, it's very small and not very active, which is why it's super important that little children are exposed to lots of germs. They can build up their immune system, literally, because after the age of 12 or so, they're no longer actively creating a mass production of T cells. They're unable to. Their thymus just isn't working as much as it was. There's the thymus. You can see it draping over that mediastinum area. Let's talk about the thymus itself. It's pink and grainy in the mediastinum, posterior to the sternum, has lobes to it, the right and left lobes. There are septa, which are the partitions dividing the lobes into lobules, and the lobules are kind of the site of where all of the action happens. You can see those lobules are little sections, right? Look like almost like a honeycomb effect. So each lobule is made of an outer cortex. The outer cortex contains the thymic epithelial cells, or TECs. Those TECs will regulate the T cell development and the T cell function. So basically determining whether those T cells are becoming helper T cells or memory T cells or cytotoxic T cells. There's also blood vessels within the cortex that help, that are surrounded by epithelial cells so that that blood-thymus barrier can be maintained. There's a central area called a medulla, and within the medulla there's no blood-thymus barrier, so this is where the T cells go into the blood vessel. This is also where the lymphatic vessels leave the thymus through that efferent lymphatic. So thymic epithelial cells form something called thymic corpuscles, and these thymic corpuscles are basically little clusters of cells in concentric layers. You can see them here, like little rings of a tree. So you can pause the video here, go back and review the section about the thymus. Let's talk briefly about the spleen. It's the largest mass of lymphoid tissue in the entire body. It performs the major function of filtering our blood. Any of the stuff in the blood that the lymph did not filter, that's what the spleen does. So it's removing abnormal red blood cells for phagocytosis. It's restoring or storing rather iron that can be recycled and reused by our red blood cells. And it's also initiating immune responses from the T and B cells in response to any antigens that are circulating in the blood. So it's a major recycling center. Overall anatomy, it's on the lateral border of the stomach, on the left side of the body, attaching to the stomach by something called the gastrosplenic ligament, basically just a big chunk of mesentery in there. We'll get into all that later. There's the diaphragmatic surface, which is smooth and convex, kind of lining right up underneath the diaphragm. There it is, that nice little purple organ on the left side. Overall anatomy, it is deep red when it's dissected. It looks like a very deep red water balloon. Very large amounts of blood inside of the spleen. Has a very soft texture, has a visceral surface with a gastric area and a renal area, just noting where those surfaces touch. And there's also a hilum. Hilum is the general term for an indentation. You'll see several organs this semester have a hilum associated with it. It's just a site of vessel entrance and exit in some cases. There's the hilum, just that kind of area of exchange where these vessels are coming into and out of the organ. Internal anatomy of the spleen, there's an outer capsule. It's very thick, has collagen and elastic fibers. It can rupture fairly easily. The spleen tissue is typically too fragile to repair, so if it is damaged, then a splenectomy can be performed where the spleen is removed. Inside, there is trabeculae. So you have this outer capsule of fibers and then this inner trabeculae, which think of the trabeculae carneae of the heart or the trabeculae of our spongy bone, right? It's this kind of spider web, stalactite, stalagmite structure where there's these little partitions that are formed, basically allowing for blood vessels to be held in place and kind of blood to kind of sit and be recycled. The pulp is the cellular components that are within the capsule itself. It's allowing for the removal of those microorganisms or any infected cells that were in the bloodstream. There's red pulp and white pulp. The red pulp has large amounts of red blood cells and macrophages gobbling up, right? Macrophages are like Pac-Man. And then the white pulp resembles the lymph nodes with lymphocytes in them there to create T cells and B cells when they're called to action. So there you see the red pulp and the white pulp and the trabeculae, which is just kind of that connective tissue network in there. This is the pathway of blood flow through the spleen. So it starts with the trabecular arteries, which are branched from the splenic artery. Then there's finer branches called central arteries. I suggest flow charting all of this out, right? It's flowcharted here, but it's very general, so be specific in your flowchart. Then the capillaries are going to take that blood into the red pulp, where there's macrophages to gobble up, like Pac-Man, and then it's flowing through the sinusoids, where there's more macrophages, and then the blood's going to collect into smaller veins and merge with the trabecular veins, which go to the hilum, which go to the splenic vein, which takes everything back out to the inferior vena cava. So you can pause the video here, and I'm going to stop the video here because I have to go, and I will continue on with this lecture in a little bit. Have a great day. Bye.